also peripheral proteins that are more loosely associated with the membrane and attached to it by bonds that can be relatively easily broken and studied. And then, yes, you can have some of these proteins will be glycosylated or lipidated that will add different functionality to them and different ways of anchoring them to the membrane. And yes, it's kind of important to remember that it's a dynamic situation. It's not static, so it's a fluid mosaic in that things are moving around, and the proteins are almost like an iceberg kind of floating in the sea, and that has implications for signal transduction that we might get to later. And yeah, you mentioned there kind of the role of cholesterol in there as well, and sterols, and the different types of fatty acids, and as we'll kind of come to in a little while as well, those kind of things can impact the fluidity of the membrane and can play important functional roles. And it's probably also at that level where dietary fatty acids like fish oils and saturated and unsaturated fats, cholesterol, statins, and those sorts of things have their impact at these lipid bilayers and can affect their fluidity and their interaction with proteins and those sorts of things. And yes, I think the next one, yes, shows it, yes, with a bit more detail, those kind of showing that difference between the integral proteins and the peripheral proteins. And the peripheral proteins can be inside or outside the cell. And yeah, polar regions and non-polar regions is kind of just another way of saying hydrophilic versus hydrophobic, or the reverse of that is lipophilic, lipophobic. So there's a few different words to describe that. And then there's also some specialized areas of the plasma membrane. Can anyone tell me what this is representing here? What's this specialized area of cell membranes?
Looks like an ion channel.
I guess technically it is.
It's a gap junction, isn't it?
Yes, you're right. Yes, so a gap junction. So what can you tell me about gap junctions?
So gap junctions are found throughout cardiac myocytes. They help distribute, or they help the propagation of action potentials between cells.
Yeah, right. And so, yeah, why is it necessary to have these gap junctions? Like, what stops the signal getting through without them?
I think the signal might be able to go through, but it takes a long time. So these help speed up the conduction. And so...
what kind of signal is it?
It's an action potential that is being transferred from one cell to the other.
Yep.
So it's usually like a pacemaker type potential.
And what is it that makes up an action potential? How would you describe an action potential in terms of the type of signal?
Action potential is a depolarization of a resting membrane potential from negative to less negative or positive.
Okay. Yeah. So the game of guess what I'm thinking is, so it's an electrical signal. And okay. So yes, electrical signals are very hard to get across a cell membrane because that's highly charged. Whereas as we talked about, the cell membrane, or the inside of it anyway, is very hydrophobic. So yes, to get that kind of rapid transmission of the signal between cells, you need these gap junctions to facilitate that. And it almost kind of makes them the two cells almost just makes them like I think the word they use is a functional syncytium, as in it's almost just one big cell because of these gap junctions that join them up. And yes, so important, yeah, cardiac muscle, and also I think smooth muscle might have them from memory. Now, this might seem like a bit of a sidetrack at this stage, but I'm curious to know within the syllabus, I'm not sure if you've had shoots or lectures or read much up on this yet. So what's your current thoughts or teaching or understanding regarding how do anesthetics work? Like, what's the mechanism of action for them? Have you had any, yes, how would you answer that question if it was asked of you?
I can give it a crack at but I might not nail it. So you've got lipid-soluble agents that appear to have their action within the lipid membrane of neurons, but also have a protein component. So there's differing theories. Yeah, it's thought to be a protein receptor and in the lipid membrane, likely.
Yeah, so that sounds pretty similar to when I was a SRMO and resident. That yes, they kind of didn't really know the answer, but it had something to do with the lipid solubility and that stuff. And I remember I don't know if you've seen the lectures by Edward Iger. I think on YouTube, I put a link to them on my blog. They're quite long, but they're very good. And he's one of the gurus of inhaled anesthetics. And yes, if you want to break from reading textbooks, watching some of those videos are worthwhile. And yes, he kind of describes this as the explanation of how anesthetics work. Yes, for many years since it was kind of first been discovered, that yes, there've been all sorts of competing theories as to how it works at the molecular mechanism. We know it works well. We can measure that with things like MAC, but in terms of the actual molecular cellular physiological mechanism that it works, it's kind of remained pretty elusive. And yes, I'm sure you're kind of, yes, the Meyer-Overton correlation for the different anesthetics, that kind of, has to do with, and so this is where some of the pharmacology comes down to the cellular physiology level, where you need to know about the structure of the lipid membrane and those sorts of things. But the reason I bring it up now is because just in recent weeks or months, there's been a bit of debate, or on Twitter at least, about the mechanism of action at this level. And so have any of you heard of the term lipid rafts recently? No, not on Twitter enough. So this is, there's some recent research on a paper released that had some results in it that seemed to suggest that that kind of postulated that they maybe solved at least a mechanism, or one of the mechanisms that might contribute to general anesthesia, and how these agents actually work at this molecular level. And it kind of relies on this concept of what they call lipid rafts. And so it's essentially an evolution of the fluid mosaic model that we talked about before. And what they found is when they looked at the cell membrane, they found what they call lateral heterogeneity in that fluid mosaic. So the distribution of the proteins and lipids and glycoproteins and other sorts of things, it wasn't random. It wasn't just diffusion and spreading out equally by Brownian motion. They found that there was some heterogeneity. So some things would bulk together, whereas other things would be more diffuse. And so the theory was that due to the different physical and chemical properties of the many different types of lipids and proteins, and plus the fact that they are free to rearrange and organize themselves, they kind of figured that, yes, that they will organize themselves into the most favorable, thermodynamically favorable arrangement in terms of that lipid solubility, insolubility, polar regions, and those sorts of things. And so what this lipid raft theory suggests is that saturated phospholipids and cholesterol, as kind of represented on that diagram, they kind of come together and form these lipid rafts in a sea of unsaturated phospholipids and other types of proteins. So so a lipidated protein is one that has various types of lipids kind of covalently bonded to it. So they're highly kind of insoluble in water. So they kind of coalesce together with these cholesterol molecules and these saturated lipids to form these lipid rafts. And yes, the recent buzz around them related to some experiments that seem to elucidate the mechanism, or at least a mechanism by which anesthetic agents might cause unconsciousness, or at least in fruit flies, which is where kind of some of the experiments were done. So it's probably still a long way off a definitive answer across everyone, but it seemed like an interesting start. So no, don't worry about that one. So yeah, this kind of explains the concept at one level here. So the blue dots on the left there, forming that kind of ordered domain, they're the very hydrophobic, saturated phospholipids, sphingomyelin, cholesterol, lipidated proteins, and so on. And they all kind of coalesce to form that ordered raft of lipids. And then the grey dots around them are kind of the less hydrophobic. They're still lipids, but they're less hydrophobic than those in the raft, and they kind of spread out into this disordered lipid bilayer surrounding the raft. And then the orange hexagons that get added in are the inhaled anaesthetic agent, and the idea is that they kind of get incorporated into these lipid rafts because they are also highly lipid soluble, but as a result they kind of cause them to expand and fall apart a bit. Now, I don't think disruption is actually a word. I think it's typo and meant to be disruption. This is from the paper that was describing it. But yes, they kind of start to spread out and fall apart a bit. And then the theory goes that, yes, in the resting normal state, you get this blue lipid raft over there with this protein phospholipase D2, which is an enzyme. And it kind of hangs out on these lipid rafts in this kind of normal state. But its substrate, which is a phospholipid called phosphatidylcholine, is not part of the lipid raft. So it's kind of away in the sea of phospholipids, and it can't catalyze or break that phospholipid down. But then the general anesthetic comes along, disrupts that raft, and as a result, this protein lipase D2 kind of translocates into the sea of phospholipids and can now act on its substrate, the phosphatidylcholine. And then the theory goes from their experiments is that, yes, once you get that anesthetic disruption of the raft, the translocation of the phospholipase D2, it can now act on the phosphatidylcholine, and it's also kind of bound to this TREC1 channel that we'll kind of talk about in a little bit, and it breaks down the phosphatidylcholine and generates this phosphatidic acid, apparently, which are those dots in red, and then it binds to the channel along with the PLD2 and opens this channel. And this TREC1 channel is a type of potassium channel, and that kind of segues nicely into the next topic. But before I go on to the next topic, are there any questions or comments at the moment? I'm not sure, sorry, how to check the comment section. So yeah, if anyone wants to unmute and ask me a question or make a comment, I'm happy to take that now.
There's no questions in the chat.
Yep. Okay, great, thanks. We'll move on. Okay, so yes, we've kind of touched on this a bit already. So the resting membrane potential. So yes, this is definitely fits into the spherical cow category. So I guess, yeah, what do we mean by the term? And is anyone keen to have a crack at describing kind of how it's generated or maintained? What are the key steps that are involved?
I can start and anyone can contribute if they want to.
Go ahead.
Probably won't know all the answers. So there's a few things that contribute to the resting membrane potential. So firstly, because of what we described already, it being like a phospholipid bilayer, that itself as a barrier does prevent particular particles or ions to traverse through it. So that'll affect the charge. For example, water can freely go across it, and so can potassium being one of the most permeable ions to go across, but it's less permeable to sodium and proteins, for example.
A generic term for that description of being permeable to potassium, but not to sodium? What the buzzword that summarizes that?
Selective permeability? I'm not sure.
Yeah, that would do. So yeah, the term like semi-permeable membrane is how it's often referred to. And that will quickly get your point across to the Viva examiner. So yes, semi-permeable membrane. So that's one key aspect of it. What are the other things that are necessary?
So there's, as we sort of talked about before, there's certain ion channels that exist there as well, and one of the main ones is a pump. So the sodium potassium ATPase pump. It's ATP dependent, and when activated, well, it's basically continuously active all the time. It'll expel 3 sodium ions out and pump 2 potassium ions inside the cell. And so that constant activity is another reason why there's imbalance of charge, creating a more negative charge inside the cell. Yeah, following on from the selective permeability about potassium, overall, because there's such an increased relative leak of potassium out of the cell, that also then means there's a relative negative charge inside the cell as well. The third thing is the Gibbs-Donnan effect, which is, I think, only a minor role, but it basically describes the fact that if you have impermeable charged molecules on one side of this membrane, then it forces the redistribution of smaller permeable molecules or ions, and so therefore the charge is again affected as well by that way. Yeah, I think, yeah, that's
yeah,
all
so
I can think of.
what is it? Why does so yeah, you mentioned the potassium leaking out through channels. So yes, why is it leaking out? What is it that the pump does that means that the potassium then leaks out? What's the key thing that's happened?
The concentration gradient?
Yes, that was correct. That's guess what I'm thinking.
Yeah.
Well, yeah. So the sodium potassium ATPase creates a concentration gradient, and that's the key thing. Because yes, as you mentioned, so the fact that it's 3 versus 2 of the different ions, that's a small contributor to the resting membrane potential, that inequality there. But the main thing is that it sets up this concentration gradient, and then the various ions will then move down their concentration gradient. So yes, it's a high potassium inside the cell, whereas a high sodium outside of the cell, relatively speaking, because of that pump. And you've already mentioned that it's more or less impermeable to sodium, so it can't move anywhere. But the potassium can, relatively speaking. So it starts to move out of the cell, and it's a positive charged ion. So then that leaves a negative charge inside the cell. And yeah, that leak of the potassium is by far the biggest contributor to the resting membrane potential. And then yes, you mentioned the Gibbs-Donnan. That seemed like a pretty decent explanation of it. Has a small contribution as well. So there's kind of, I guess, yeah, I think that the 3 main things that in an exam you mentioned would be, yeah, the biggest one is that establishment of the concentration gradient, and then the leak of potassium out of the cell. That's the biggest contributor. And then the 2 smaller ones are that generated by the ATPase itself because of the unequal nature of the pumping of the ions, and then the Gibbs-Donnan as well. And yes, again, yeah, it's important to not think of this as, again, it's called the resting membrane potential, but that's really only in contrast to when it gets activated by an action potential or something. But even in resting cells, the cells are anything but at rest. So it's kind of very much a dynamic equilibrium that they reach at this resting membrane potential, and the sodium potassium ATPase, as you said, is constantly going. So there's a lot happening when nothing's happening. So it's important to remember, yes, that it's a dynamic equilibrium of ions moving in and out. And so, yeah, so what is it that eventually stops, you mentioned kind of the potassium leaking out. Why doesn't it leak out and completely dissipate that gradient? What else do we need to consider here? We've talked about the concentration chemical gradient. What's the other gradient at work?
The electrical gradient.
Electrical, yeah. So yes, they're often combined to the electrochemical gradient. Let's have a look here. Just checking my notes. Actually, yes, we might go on to the next slide. So, so this is just kind of a summary slide. So yes, the three main points there. So yeah, semi-permeable membrane, the concentration gradient generated by the sodium-potassium ATPase, and then the ions move down their concentration gradient. I don't know why I've got a question mark at the end of that point. So yes, just going through my notes again, semi-permeable ATPase, yes. Yes, and the other thing to remember that this the membrane potential is really only a thing, yes, at the very edge of the plasma membrane. So the cytosol and the extracellular fluid are pretty much electrically neutral, but yes, it's just this very thin section either side of the membrane itself that has these positive and negative charges. And yeah, so does anyone have any numbers in their head for what the resting membrane potential value is and what the intracellular and extracellular concentrations of the important ions are? Depends on what textbook you read or what cell you're talking about, but do you have any ballpark figures?
Give that a crack. I think the resting membrane potential in a nerve cell is minus, I think it's minus 90. And the Nernst equation for potassium is minus 94.
Right.
And there's heaps and heaps of potassium inside, I think 150 millimoles per litre versus 5 in the extracellular fluid.
Yeah. You mentioned Nernst, now you've done it. What can you tell me about Nernst and his friend?
So the Nernst potential looks at each individual electrolyte, and if it was the sole electrolyte in the system at its electrochemical balance between the gradients pushing it in and outside of the cell at equilibrium, what membrane potential would be produced for each single electrolyte.
Yeah. And do you know what that's called for each?
I think that's the Nernst potential for each one. And if
that's
you can
probably the more technical term. Yes, you're right. And then I think just equilibrium potential is the kind of, yes, the generic one. And then, yeah, so what's Nernst's friend's name? You know,
so there's Goldman Katz.
Yeah, there's probably a whole stack of people that didn't get recognised for their work on the project. But yeah, so the class one is the Nernst-Goldman equation. And so what does that involve?
So that's essentially the electrolytes sodium, chloride, and potassium, their Nernst potential essentially put together in a single equation.
Yep. Yep. No, that sounds like you got a good grasp of that. So this is from Rang and Dale. And so this is just a kind of a stylized random cell. So as I said, depending on which textbook you read or what cell you're talking about, there will be different values, but there's some general ballpark figures, and it's much the, as long as you get them in the ballpark, you probably won't even get asked this kind of level of detail, but I think this picture sums up what we're talking about pretty well. So it's got the sodium potassium ATPase there, showing the sodium and potassium being pumped out these resting potassium channels that provide that constant leak of potassium out of the cell. And then a couple of other exchanges and channels that kind of fit in as well. And then it's got that equilibrium potential down the middle for the various ions that we're interested in there. Now, actually, I might just ask that now. So what can you tell me about the chloride values for chloride there? Like most of the discussion, kind of understandably, is about sodium and potassium, but chloride certainly has a role. What can you tell me about the role of chloride?
So this is showing that there's a high concentration of chloride extracellularly And if it was able to freely flow into the cell, it would pour in, creating a negative 90 resting membrane potential.
Yeah. And so are there times where we mess around with, so yeah, they talk about the kind of conductance of the membrane, like through opening and closing these channels. So are there times where we mess around with chloride conductance or the chloride permeability?
So propofol, or when a GABA receptor is stimulated, opens the chloride channels to allow chloride to rush in, and then you'll hyperpolarize your cell so it won't transmit. Yep,
that's right. Okay. Yes, so the goal, but yeah, so actually we've talked about this already a bit. So yes, just the Goldman equation. Yeah, anyway, yeah, so what can you tell me about this equation? What what's it about? What are all those? You can probably ignore the bit at the beginning, and actually just out of interest, the F there is the Faraday constant. I think in Blair Munford's shoot on electrical stuff a little while back, he talked about the Faraday constant. So it's come up here again, and that's to do with the charge of an electron and stuff. So don't need to worry about all that constant in the front. But yes, that bit in the brackets. So what's that kind of describing there?
So I think that's the concentration of the so first the potassium and sodium outside the cell, and then the chloride inside the cell over the opposite. So the potassium, concentration of potassium and sodium inside and the chloride outside. I think the reason the chloride's flipped is something to do with its valency, and I don't understand much more than that. Yes.
So what is the difference between chloride ions and sodium and potassium ions?
Not clever enough at chemistry to understand that. But it's to do with not its charge, but it's I can't remember.
Well, no, I think it is its charge, and so I think that's why they're flipped around like that. But I think where the valency comes in is with something like calcium. So yes, I think the way the Goldman equation is set up, it has the positively charged ions on the top and the negatively, well, in terms of the outside or inside is determined by what their charge is. Now, if it's a ion that has a 2 charge, so calcium 2+, if you're going to include calcium in the Goldman equation, you need to put a 2 in front of it to account for its valency there. So I think that's where that side of thing comes in, but you may want to double check on that. But that's my understanding of it. Anyone else have thoughts on the Goldman equation and how the charges and valences and negative, positive fits in?
Can you or anybody give me an easy way to remember the one that's on top versus the one that's the bottom? I can never remember outside versus inside or know if it's at all important to, do I need to know that? Or how can I remember it?
My memory, at most, it could theoretically come up in a multiple choice. I don't even know if that's likely. I can't imagine them asking you to recreate it in a Viva setting. But yeah, I don't know if I had any special mnemonic to remember which way round it was. I know that, I always knew that, chloride was different, but I didn't know whether it was outside, on top, or inside. They're not even in alphabetical order, are they? The O wasn't the I, or I'm not sure. Anyone else got any tips?
Yeah, and do you just mean like how to remember if it's the extracellular concentration on top or the?
Yeah.
I don't know. Maybe you can just think of like if you're using that kind of notation, then O is for on top.
There you go.
Winner.
That's chloride, and chloride's the opposite. Yeah. O on top. Yeah. There you go.
Chloride's the exception because it's negative.
Yeah.
And I is inferior. I love it.
So, okay. So, yes. No, we kind of covered what I wanted to say about that with the chloride permeability. Oh yeah, so now with the Goldman equation, so there's actually a simulator where you can actually play around with this now. It used to be online, and so I'd hope to bring that up, but it's been taken off the website. You can download a version, but that only works on PC, so it doesn't work on the Mac. So I can't show it there, but I'm going to try and show it to you. There's a iOS version, so I'm going to try and show it to you on my iPad, but this is where everything could go horribly wrong. So just bear with me if I can. How do I stop the share? Why can't I see my... So is Ben able to stop the sharing of my screen? Because I don't seem to be able to do that.
Are you able to click the share screen in the bottom of your, on the bottom part of your screen in the middle?
The problem is I can't see my mouse at all at the moment. So it's not really, I can't really select anything. So it's going to the next screen, which is, might have to skip that then. But yes, yeah, anyway, you kind of see from the screen there the sorts of things you can do where you can play around with the potassium and sodium intracellular and extracellular concentration, as well as the permeability to them. And you can see what effect that has on their equilibrium potentials and also the resting membrane potential as well. And then there's some presets that you can have for different cell types, like a red cell or an axon. And then, yes, you can then do things with it where like you could kind of simulate an action potential coming through where all of a sudden the sodium conductance increases and see what happens there. So yes, if you're kind of a visual person and like playing around with those things, that's worth getting hold of. So it's in the App Store, at least for iOS. I don't know about Android. But again, I think on the blog site, there's some links to where you can access that. And I'll probably need to update them now that that website's kind of gone down. But yes, that can help you get a more visual kind of dynamic image of what's going on with those potentials. Gibbs-Donnan, we kind of covered that already pretty well, I thought. And yes, that's kind of a graphical representation of it. So yes, altering the distribution of diffusible cations and anions where there's a non-diffusible one. And what is the non-diffusible kind of ion that we're mostly talking about when we're talking about Gibbs-Donnan?
The protein.
Protein. And yes, what's the predominant kind of average charge of proteins that we're involved in the cell stuff?
And negatively charged.
Yeah, tend to be negative, that's right. And then there's the Donnan ratio down the bottom there. Again, I don't know that you need to know too much detail about this, probably mostly MCQs and just knowing that it contributes to, well, there's a few things that, so as well as a resting membrane potential, there's a couple of other things where it gets a mention. Can you think of anything else where the Gibbs-Donnan effect or equilibrium or ratio plays a role? Sorry, what was that?
The oncotic pressure
Yes.
is larger in practice than you would assume by calculating it.
Yes, that's right. Yeah, so the plasma oncotic pressure, yes, in capillaries and that sort of thing, I think. And then there's one other one as well. So the cell volume, I think it has a role there. And I think this is where it kind of interplays with the sodium potassium ATPase a bit, I think. I think it might be that without it being counteracted by the in equal charge movement with the sodium potassium ATPase, that eventually the Gibbs-Donnan effect would lead to the cell swelling up and kind of lysing. So it needs to be kept in check, and part of the way that that's kept in check is through the sodium-potassium HCPAs, because if you've got those the negative ions inside, then yes, it will redistribute things, but then that then changes the cell volume. So then that upsets that equilibrium again. So it keeps this positive feedback loop until if it was left unchecked, I think the cell would swell up and lyse. Now, I think I've got that the right way around, and then it's other mechanisms like the sodium potassium HCPAs that counteracts that. But they're the three situations that I'm aware of where it plays a role: cell volume, resting membrane potential, and the plasma oncotic pressure. And I'm not sure that you need to know too much more than that. So I guess, yes, given what we've just described, we can kind of go back to this lipid raft model of the molecular mechanisms for general anaesthetics and think about what might happen here. So if we've got this disruption of lipid rafts, then the translocation of the phospholipase D and binding to the TREC1 channel and the phosphatidic acid being produced and opening it, and we know that it's a potassium channel. What would you predict would be the effect of opening that TREC1 channel?
Would you get loss of potassium outside of the cell? Is that what you're alluding to?
Yeah, so you'd get potassium moving down its concentration gradient outside of the cell. Now, given what we've just discussed about the equilibrium potentials and the resting membrane potential, what effect is increased movement of potassium from inside to outside going to do to the resting membrane potential?
So potassium efflux out of the cell tends to make it more hyperpolarized and more negative.
That's right. And so you can kind of see how that might induce anesthesia in someone. If it's going to, so anything that hyperpolarizes the membrane, it's going to make it less easy to depolarize and trigger an action potential. So it's plausible that, yes, this could be a mechanism for how general anesthetics could induce anesthesia. And it's probably not the only one, and the authors kind of say that. But yes, there's kind of, they involve kind of knockout fruit flies and various different mechanisms to draw this out. But I thought it was an interesting example of, yes, kind of seeing how these different aspects of the plasma membrane and the resting membrane potential can interact in this context of general anesthesia. Now, it's probably, very unlikely this is going to pop up in the exams anytime soon, and it's still kind of early days, but I thought worth kind of including as much just for the teaching points about the other aspects of lipid membranes and resting membrane potential. But it's one to keep an eye out in the future. Oh yes, and also again on the blog site is a link to this paper, which I think does a pretty good job of explaining the generation of the resting membrane potential. So that's worth having a look at as well if you need a refresher or didn't quite get it this time round. Any questions at this point? Again, can't get my mouse to see any questions in the comments, but if there's any. No. Okay. So the next question. So yeah, we've talked about this membrane, and yes, some of the challenges of getting things across it. So yeah, how do we get things across the membrane? What kind of things do we want to get across it, and how do we achieve that?
So if you're looking at things that you want to transfer from outside to inside the cell, so there's a few factors that determine whether or not you're able to do that easily. So the membrane is a phospholipid membrane, so it's able to transfer things which are lipid soluble because it's a lipid. Sorry. And then you're thinking about things that are ions, for instance, need ion channels, and if it's anything that's quite large, it maybe need to be enveloped in the membrane through pinocytosis or something, and then transferred intracellularly. Yep.
Okay. So yeah, what are the mechanisms that you're aware of that the cell has at its disposal to get things across that fit into those different categories, and how is it achieved?
Okay, so lipid-soluble things such as lipid-soluble drugs can transfer across themselves. So they're things like local anaesthetics or opioids.
So what's that process called?
Don't know. Is it just diffusion?
Yeah, pretty much. So yes, and that's where Fick's equation all comes in, and kind of that's something you kind of keeps popping up in the exam. So that's certainly worth knowing, because as you said, yes, it's kind of can be pharmacology about getting local anaesthetics across and into the membrane, and concentration gradients and those sorts of things. So diffusion's definitely a big one. So you mentioned some of the anaesthetic agents and the local anaesthetics. There's two other super important things that we're very interested in that get across cell membranes by diffusion. You know what? Anyone have a guess at what they are?
Oxygen and carbon dioxide.
Yeah, that's right. So I think for a long time they thought that oxygen was actively pumped across from the lungs into the blood, but no, it's just by pure diffusion, but over a massive surface area in the lungs. So yes, both oxygen, CO2, and a number of drugs are just by diffusion. So yes, what are some other mechanisms?
So another mechanism would be ion channels.
Yes. So how do ion channels work? Not so much some of this we'll cover in more depth in, so I think I've got another tute later on nerve and muscle physiology, and also, some of the other ones covering in a bit more depth. But effectively an ion channel, how would you describe how they work? Or what's the process that's involved in that?
Well, a lot of the channels that we talk about are ligand activated channels. So you have binding of something to an extracellular site on the channel, which then causes it to open.
Yep.
And then you can get transmission of electrolytes or ions when the ion channel has gone from like sort of a closed to an active open state.
Yeah. And so what is it that drives the movement of those ions through the channel?
It still uses diffusion, so it still travels down a concentration gradient.
Yeah, that's right. So there's this other kind of broad category of, so while there are some things that can just get across by diffusion, they're lipid soluble enough to do that without any need of any kind of transporter or protein. There's another category called facilitated diffusion, where it's molecules that are still moving down a concentration gradient or an electrochemical gradient, but they can't get across the membrane by themselves, or we don't want them to. So there's some other mechanism that needs to be invoked. And so that could be some kind of ion channel or some other transporter where it just uses its own concentration gradient as the driver for it, even if there's some exogenous trigger that might activate or open that mechanism. It's still, at the end of the day, just diffusion. And what are some other kind of broad mechanisms like that? So I've talked about diffusion, facilitated diffusion.
Is this still for me, or is someone else?
Oh, anyone can jump in.
There's primary transport, like active and secondary transport.
Yes. So yes, can you elaborate on them a bit more?
So active transport is a form that requires energy. So like hydrogen ATPase or the sodium, potassium ATPase are examples of those.
Yep.
And then secondary, it's a bit harder to explain. It's kind of like so it uses the transportation of the molecule is relying on the concentration gradient that's being developed that's been established by a different protein that uses the active transport mechanisms. So the classic example is like in cardiomyocytes, you have the sodium potassium ATPase pump, and then that gradient that develops sets up the activity for the sodium calcium exchange, and then that allows for contraction to happen in the cardiomyocyte, and that's how digoxin is meant to interfere with that.
Yep. No, that sounds like a pretty good description there. So you've got yes, primary active transport, where you're using ATPase directly to move things against their concentration gradient. And so the sodium potassium ATPase is a classic example of primary active transport. It's hydrolyzes ATP and pushes both sodium and potassium against their concentration gradient. And the secondary active transport that you should mention, yes, so that's where it kind of, yes, it will move something against its concentration gradient by utilizing the concentration gradient of some other species. And so, yes, the most common one is sodium, because there's a huge concentration gradient for that to kind of move into the cell. So you can pump things out of the cell by allowing sodium back in the other direction, and there's other ways it can be done as well. And another example of that is the SGLT co-transporters. So they'll move, I think it's kind of sodium down its concentration gradient and move glucose back up its concentration gradient. And that one's got a bit of press recently as well because of the SGLT2 inhibitors and the issues they cause around sick and peri-op patients. And then kind of mentioned exocytosis and endocytosis, although often that's not necessarily getting them across the membrane. It's often just covering them in another membrane, but a way of getting things into and out of the cells. Now I think I've got a slide yet. And so this one is from Ganong, actually, which is a common textbook for the primary. And yeah, that kind of, yes, it's kind of overwhelming to begin with, but it shows some of the different types of transports and gives some examples of them. So up the top there's the active primary transport, where you've got the sodium potassium ATPase. And then, yes, it's even kind of mentions an inhibitor of that. And then co-transporters on the right-hand side there, and then the counter-transports. So they're kind of the secondary active transports, I think. So yes, that can be one to come back to for just some summaries of the different transport mechanisms. But yes, the key ones to remember, I think I've got it on the next slide. So diffusion, facilitated diffusion, primary active transport, secondary active transport, and then sort of exocytosis and endocytosis. Any other questions or comments at this point? No. Okay, so we've talked about getting kind of physical things across the membrane, but what about getting signals across the membrane? So we kind of talk briefly about how hard that is to do and how gap junctions can play a role in that in some cells, but what are some other ways that we can get signals across membranes? And the generic term of signal transduction is what they call it. So yes, and we've touched on a couple recently, but what are the ways we have of performing signal transduction?
So you've got protein-gated, G-protein coupled receptors and ligand-gated voltage channels.
Yes, yep. There's certainly two big ones that we need to know about. There's a couple of others, at least.
I think tyrosine kinase.
Yes.
One.
Can you think of kind of, there's lots of them, but there's kind of one example that's kind of relevant to what we do in some sense?
There's voltage-gated ion channels as well.
Yeah, I was thinking of, yes, Can anyone think of a tyrosine kinase linked receptor that we play around with a bit?
The insulin receptor.
Insulin, yeah. So that's, yes, if you want to learn about the tyrosine kinase receptor, that's the one to look into. I don't know that you'd like to get asked much on it, but that's one that's relatively well characterized and at least relevant to our practice in some sense. So we've got ligand and voltage gated ion channels, G protein coupled receptors, tyrosine kinase receptors, and then there's probably one other big category that we need to know about as well.
I think that's receptors at the nucleus.
Yes, that's right. Although, yes, technically some of them are in the cytosol and then move into the nucleus, but yes, that's probably, yeah, nuclear receptors or the kind of the steroid type hormones. And so a lot of this will come into the endocrine tube that I'll give later, so we won't go into too much depth today, but yeah, one that does kind of come up in the primary from time to time is the G protein coupled receptor. So maybe I'll go back. What can you tell me about G protein coupled receptors? How would you describe them, or what are the key features? What does the receptor look like? How would you describe the receptor itself?
So it's got, I don't know what they're actually called, but like 7 sort of linked subunits that make up the receptor. So something will bind onto that receptor, and then it will activate second messengers, which are inside the cell membrane, and that depends on the type of G protein coupled receptor. So there's the GS, GI, and GQ, and they will all have slightly different, I guess, not reactions, but responses inside the cell. So the GS will increase cyclic AMP, and then the GI will decrease it, and then the GQ, I think, increases IP3, and then they will activate the second messengers. Yeah, well, those are the second messages, which will then go on to lead to their effect. Yep.
And
yeah,
you know, so you're right. There's kind of 1 or 2 steps you've missed out. Yes. Can anyone fill in the gaps on those other steps? So you've got the ligand binding to the external outside of the receptor and a conformational change, and then what are the steps that then lead up to the generation of those second messengers?
There's adenyl cyclase.
Yes. Yep.
Or the guanyl. I can't say it. That one. Guanyl
cyclase. Yes.
Yep. And then they, depending on which one it is, I can picture it, but I can't describe it. They will sort of activate, and then it's on the inside layer of the membrane. It will kind of shift the position, but I just can't remember exactly how to describe it.
Yep. So what does the G protein in G protein coupled receptor, what's that referring to? Anyone, doesn't have to be you, Annabelle.
Yeah, you go for it.
I'm not sure, but with the, is it the gamma subunit of the alpha beta gamma combination? Or it could be the GTP. I was thinking. I don't know.
Yep. Yes. No. Yep. You're on the right track. We'll go to the slide, and yeah. So this is kind of a decent summary slide once you can tease it apart about how the various types of G protein coupled receptors act. So yes, you've got I think they call it a serpentine receptor or something, because it's got all, is it 7 transmembrane domains? So kind of going back and through, it's like one long protein as opposed to kind of a subunit receptor like and channel that some of the other ones are. So it's this 7 transmembrane domain proteins, and then all sorts of different things can bind to it extracellularly. But then, yes, it's the intracellular stuff that we're most interested in. And so, yes, the G protein refers to those, the alpha, beta, gamma subunits that kind of coalesce inside the plasma membrane. And yeah, this is where that fluid mosaic model comes in again. So they're kind of peripheral or peripheral proteins. I can't remember that. So they're loosely associated with the plasma membrane. They're not integral proteins, but they kind of can move around on the plasma membrane and can come into close association with the G protein coupled receptor. So they're right there when it gets activated. And then, yes, once it gets the conformational change, you get release of the alpha subunit, and then the beta and gamma subunit go off and have some effects, and the alpha subunit goes off and has some effects as well. And it tends to be the alpha subunit that we're most interested in. However, the beta and gamma subunits also play a key role as well. So what can you tell me about the alpha subunit there? You kind of mentioned GTP before. What does that GTP bit involve? And that's where the G protein comes from, the GTP that's involved.
Yeah, so I think I always get this mixed up, but so I think, so as the picture sort of alludes, when it's when the whole receptor complex is activated, then the alpha subunit will bind to GDP, separate itself from the beta and gamma. The beta and gamma stay together as like a dimer, and then has its own sort of separate effects elsewhere along the cell membrane. The GDP then gets undergoes dephosphorylation and then becomes GDP and the phosphate, and then that leads to the all the second messenger stuff that Annabelle describes.
Yep, that's pretty close. Except probably the key thing to remember is that when it's together as the alpha, beta, gamma subunit, it's has GD. I'm trying to think now. Yes, I think it's GDP that is in the alpha subunit at that point, and then
okay.
Yeah, you might need to double check this, and it'll be in a bit more depth in the endocrine shoot, I think. But I'm pretty sure it's GDP that's in there. When you get that conformational change, the GDP gets released. GTP moves into that spot instead, and that frees the alpha subunit from the other two. And yeah, the beta and gamma subunits go off and do their thing. And interesting, you'll notice there that you might have heard this, that one of the enzymes that it activates is a phospholipase C. So we talked about before with general anesthetics, perhaps working via a phospholipase D mechanism. So there's some similarity there. And these beta and gamma subunits, yeah, they stay tethered to the cell membrane there. But yes, the alpha subunit, now that it's got GTP in it, it goes off and then activates the downstream things and activates adenylyl cyclase and protein kinase A and all these downstream effects. And then what is it that switches off this signal? So you've got the alpha subunit with GTP bound going about happily activating all these downstream things. What eventually turns it off? What does it need to do to get back to the resting state?
I know you've got arrestin, which sort of blocks the process, but I think the GTP sort of loses its phosphate and turns back to GDP.
Yeah, that's right. Arrestin, that's a new one to me. Might be right. Maybe I'll have to look that up before the endocrine shoot. But yeah, so the key thing about these G proteins is that the alpha subunit has GTPase activity within it. So we're most used to ATPases, like the sodium potassium ATPase, but there's also GTPases. And these alpha subunits have intrinsic enzymatic activity to turn GTP into GDP. And it's that activity that switches it off. So when the receptor is activated, the alpha subunit gets rid of the GDP that's in there, binds GTP and goes off and does its thing. And then the thing that determines the duration of that signal is the enzymatic activity or the kinetics of that GTPase. So that signal will stay on for as long as it takes to hydrolyze that GTP into GDP. And then once it's in GDP, it goes back, binds the beta and gamma subunit and deactivates them and goes back to that resting state. And interestingly, not so fun fact, it's this G protein mechanism that where cholera toxin kind of stuffs things up, and it kind of blocks that GTPase activity. So the G protein remains constitutively active and then goes on and causes downstream effects in the gut that kind of results in that nasty diarrhea. So that's kind of some clinical relevance of this, not necessarily to anesthesia per se, but anyway, yeah, so that's not a bad slide to come back to. And as I said, we'll go into that in a bit more depth, I think, in the endocrine shoot. I think on that old syllabus thing, there was something about the list of organelles. So the organelles are the little things inside the cell. Can you think or list any that you know of? Again, I think this is fine print stuff. Might scrape into a multiple choice question. There's one organelle that might be more relevant than the others, but there's quite a few.
So mitochondria,
Yes.
and then endoplasmic reticulum.
Yep.
Nucleus.
Yep.
I'm out now.
Well, you got the main ones probably. Yeah, I won't kind of labour the point too much. There's a whole stack of them. Some, yes, kind of better understood than others. But yeah, certainly if you're going to be asked about any of them in depth, I guess there might be some questions related to the nucleus with that kind of signal transduction and protein production and transcription, that sort of stuff, but I don't think so. But yeah, the mitochondrion kind of comes into it a bit, and that's a kind of a electron microscope picture of it there, often described as sausage shaped, and you can see why. And oh yes, there's yes, the jokes tend not to work as well over Zoom. But yeah, what can you tell me about, what is it? Oh. No, my slides are not working well. Anyway, what can you tell me about the mitochondria? Just give me, I'm going to try and bring my notes up again. I'll need to stop and then play again.
So the mitochondria is where you have the Krebs cycle and then oxidative phosphorylation producing energy.
Yep. What can you tell me about its structure? How's it kind of set up to do that?
It's got a bilayer where there's a high concentration of hydrogen ions for the oxidative phosphorylation process.
Yep. Yep.
And then you've got proton pumps to pump hydrogen into that. And then as hydrogen flows back out of that, it stimulates the production of ATP.
Yep. Yep. Yeah, so they talk about, and you can kind of see some of the structural features there. So there's, as you say, the kind of the membrane bilayers, and there's kind of two of them. So there's an outer membrane, and then an intermembrane space, and then an inner membrane. And so the inner membrane and the outer membrane, they're both phospholipid bilayers. So it's kind of like a double bilayer in a sense, but then there's this intermembrane space as well. There's kind of invaginations that you can kind of see there called cristae that are designed to increase the surface area available for all the reactions and the oxidative phosphorylations going on. And then there's the matrix as well, which is kind of the inside, which is the cytoplasm effectively, I guess, of the mitochondrion. And then there's another quirky thing that's contained within the mitochondria, which is a little bit weird. Unusual. Do you know what that is?
Are you referring to those little dark spots?
Well, I don't think so. I don't know what those dark spots are, but
the only thing I was going to say is that the mitochondria has its own genetic material, which is separate from the organism, other organisms hosting it.
Right, and interestingly, I think it's actually a circular DNA rather than a strand DNA, and I think the theory goes that at some point in the deep dark history of evolution, the mitochondria was a bacteria that kind of ended up being kind of in a symbiotic relationship with whatever other animal or creature it was, and it's kind of, yes, become an essential component. And yeah, that circular DNA, which is characteristic of bacteria, is kind of one suggestion of that. And also, you get all your mitochondria from your mum, so that kind of mitochondrial DNA has a maternal inheritance pattern. And there's also some diseases related to mitochondrial DNA that can kind of get inherited through that as well. But again, I think that's unlikely to be an issue related to the primary exam. Although I think from memory, some of those propofol infusion syndromes and stuff might be related to metabolism of propofol and mitochondria in kids and issues with that. But I'm not sure how likely that's going to come up. So yeah, started talking there about the electron transport chain. Yeah, this is kind of a diagram of it. So kind of starting over on the left side there, you've got NADH, and that kind of donating some electrons to the beginning of the electron transport chain and ending up back as NAD+. And then kind of as these donated electrons kind of move through all these different proteins, you kind of start ejecting hydrogen ions out into that intermembrane space. And so it's a very narrow space, and that's why you get a very high concentration of hydrogen ions in there. And you can see the various places where the hydrogen ions get ejected, and then towards the end, you get, this is where the oxidative bit comes in, you get oxygen coming in, and it gets reduced into water. And so yes, you may have already been aware of this, but this is the interesting bit where you realize that the oxygen you breathe in doesn't get turned into carbon dioxide that you breathe out. It gets turned into water, and the carbon dioxide kind of comes from the nutrients that you metabolize. So yes, you end up with water, and then as you mentioned, yes, there's the ATP synthase, the hydrogen ions kind of move back through that kind of fascinating mechanism to generate ADP, and that whole process is called oxidative phosphorylation, because you're phosphorylating ADP into ATP, and it's kind of oxidative because yes, you need that oxygen to get reduced into the water to as a crucial part of that, because if you haven't got that oxygen to do that, then that kind of backs up that whole process and kind of slows it down. And so it's they talk about this whole process being coupled. So you can't produce ATP with this mechanism anyway, unless you're getting oxygen to it. And similarly, you also need this the NADH at the beginning as well. So if you don't have the oxygen and you don't have the NADH, then this process is going to shut down because you haven't got the mechanisms to do that. And that process is what they call coupling. That process is coupled. And then have you heard of the term of uncoupling things, or uncoupling proteins? Any of that ringing a bell?
So that's in brown fat thermogenesis.
Yeah. So non-shivering thermogenesis. And so yes, in brown fat, there's something called uncoupling protein, or thermogenin. And it kind of shortcuts that process. It uncouples it. And so that provides a way to dissipate that hydrogen ion gradient without needing to produce ATP. Because that's right, the other thing is if you haven't got any ADP, it will stop as well. So generally it means that you can't utilize this mechanism unless you're burning energy somewhere. You need to be using up ADP to provide it as a substrate for this mechanism. And so that's why normally when we get cold, we shiver, so that we're using muscles and we're consuming ADP, and then that will generate this process that will produce extra heat. But in brown fat, you don't need to shiver, because you can dissipate that gradient, keep all the other metabolic processes operating that generate heat without needing to actually move any muscles and use up oxygen as much in muscle contraction. So that's that uncoupling protein. There's a couple of other things that can effectively do the same thing. So cyanide poisoning works in the same way by uncoupling that process, and then you kind of, but it does it in a way, that is unhelpful and doesn't produce any ATP. And so that's a bit of a nasty one. And then there's also this one that apparently is popped up as a weight loss supplement. So I think trying to replicate the effects of brown fat by burning fat and calories without actually having to do any exercise. And I don't know if you've seen it, it's called dinitrophenol, and it acts as an uncoupler, but unfortunately its therapeutic range is tiny. And so it's very easy to overdose. And if you do, there's basically no antidote to it. So you're pretty much destined to die. And so I've not seen it myself, but I've heard stories of people turning up in ED. And as you can see, it's a pretty yellow color. And if they've overdosed, then that color ends up in pretty much every body fluid they have. So they're crying and sweating, and everything coming out of them is kind of bright yellow color. So that's a bit of a red flag or yellow flag, I guess, to why they might be so overheating and agitated and about to die. How are we going for time? Okay, what's next? Any questions at this point? We can go back. Maybe. Is this? Yeah, if there's any questions about that side of things, and we'll kind of go back on and look at the steps leading up to this, like where the NADH comes from. And again, there's another tute I give is nutrition and metabolism. So we go into a bit more depth about this side of things in that tute as well. Any questions at this stage? Okay. Okay, so just let me go back one. Actually, maybe at this question, yeah. So yeah, this is kind of oxidative phosphorylation, and one question to possibly ask yourself that, yeah. Well, I'll ask it now, but yeah, as this unfolds, and certainly in the nutrition and metabolism tube, we'll cover it in a bit more detail. But this is kind of oxidative phosphorylation. Now, when we go into anaerobic metabolism, we produce lactate. And so, yeah, I guess one question is, yeah, does anyone know why lactate is produced when we're kind of in that anaerobic state with either not enough oxygen or our metabolic demands are outstripping what we can supply in terms of oxygen?
Is it so it's a secondary process of fermentation, so it tends to reduce NADH, NAD to NADH, so it can be fed back in through the glycolytic pathway?
Yeah, that's right. So you can see here and through other processes. So yes, NADH is a key molecule in all these metabolic processes. And as we move back up to the metabolic processes that produce this NADH, it's important to be able to recycle that NAD that gets used back into NADH. Do you know what the precursor is that ends up as the lactate?
So pyruvate tends to turn into lactate.
Yes, that's right. So I think we'll work our way back up to that as we go along. So I think this next one is, yes, so this is the Krebs cycle or the citric acid cycle. It's got a few different names. Yeah, so this is just kind of going back up one step, and yeah, kind of shows, yes, it's a bit overwhelming. It shows, yeah, again, the crucial role of NADH, NAD being produced in this place. And so this is a very big source of the NADH that is used in that oxidative phosphorylation process. So you can see there's a number of points there where you produce that NADH that then gets utilized with the electron transport chain. There's also, yes, the production of one GTP there, which is a bit interesting rather than ATP. But in some ways, the GTP and ATP are interchangeable. Like there's enzymes that can convert GTP into ATP. So that's when they I don't know if they still ask those questions about how many ATP molecules are produced from one glucose molecule, et cetera, and the numbers vary a bit. But I think they consider that GTP as an ATP molecule because they're kind of interchangeable. And also, yes, this shows where the CO2 is produced that we breathe out. And so, yes, just that it's emphasizing again that it doesn't directly come from the oxygen that we breathe in, even though that seems the simplest explanation when you look at what we breathe in and breathe out. And yes, the other key molecule here that's the acetyl-CoA. So there's kind of a slide at the end that shows the central role of acetyl-CoA. It keeps kind of popping up everywhere and is a key regulatory domain. But I don't think you really need to know this in detail. I think in the US, it's kind of a running joke that you need to know this for your USMLE or whatever, and then you can forget it straight away. So I think that the key things to take home from this is that it's a big source of NADH, and it also gives you a little bit of ATP as well, and that it's the acetyl-CoA that's kind of being used to kind of kick all that off. Yes, and yeah, this is talking a bit about that. I think the Cori cycle they call it, where you get glucose down into pyruvate, and I think we'll have the glycolysis slide is next. And then, yeah, this is the key step here. So we've just talked about the Krebs cycle. And then, yes, kind of very much shortcutting it then through oxidative phosphorylation. But we've got that acetyl-CoA that was in the previous slide at the beginning of the Krebs cycle. But then kind of going back up through that, you've got this key step here with pyruvate dehydrogenase, where you've got the pyruvate and the acetyl-CoA. Now, that step going from pyruvate to acetyl-CoA is a key step because it's irreversible. That's why, so from once you've got acetyl-CoA, so I'm kind of getting ahead of myself a bit here. And again, if you're around for the metabolism tubes, we'll cover that a bit more. But one of the key things about pyruvate is you can turn it back into glucose. You can turn it back into a carbohydrate. And as I'm sure you're aware, they're crucial in terms of feeding the brain in particular. So you don't want to turn pyruvate into acetyl-CoA lightly, because once you can't turn acetyl-CoA back into glucose, so you want to make sure that you're only doing that pyruvate to acetyl-CoA step when you really need to. And then, yes, we also talked about regenerating the NADH by converting pyruvate into lactic acid, so that, and again, we'll come to this in the the glycolytic cycle as well, that you need NAD plus for that. Yeah, so that's basically just showing the Cori cycle. And yes, and again, yes, going back up through gluconeogenesis into glucose as well. So the key things from this slide are that pyruvate to acetyl-CoA is a key regulatory step that's irreversible and commits pyruvate to going to acetyl-CoA instead of being able to be converted back into glucose. And then there's also this anaerobic metabolism of pyruvate to lactic acid when you have the shortage of that NAD+. I think the next one, yes, again, so a little bit crazy, but yes, so this is glycolysis. And the key things from this are that one molecule of glucose kind of gets split into two of the three molecule things. So that's where counting the ADPs can get a bit tricky. But again, you'll see that what the key, so this glycolysis, it doesn't use oxygen. So this is all you've got when you've got no oxygen, because you need oxygen for oxidative phosphorylation. But when you're in that anaerobic state, this is the only place you can get extra ATP from. Doesn't produce heaps, so you need a lot of glucose to do it. And you can see in the middle at step 6 there, that's where you have that NAD plus going to NADH. So if you run out of NAD plus, then even glycolysis grinds to a halt. And so that's why you then need to go back and convert the pyruvate. So you end up down at after step 10, you've got pyruvate, but you need to convert that into lactic acid in order to get the NAD plus back, so you can keep the glycolysis going. And that's where the lactate comes from. Is there anything about that? Probably not. Yeah. So the main thing are that, yeah, so glycolysis, the 6-carbon molecule ends up as 2 3-carbon pyruvates, generates ATP along the way, and you need to have NAD plus and a phosphate, which normally isn't an issue unless you're severely malnourished, to keep glycolysis going. And as I said, yes, this is all you've got when you're in anaerobic conditions. Now, this is a little tangent, almost literally, from glycolysis. Has anyone heard of the Rappaport-Lubring shunt in the context of glycolysis?
Nope.
No. Okay, so this is, again, probably not examinable, but it's just a point of interest because it has some kind of relevance to things that we're interested in. So the Rappaport-Lubring shunt, it's a, I'll see if I can work out where it comes off. I think it's this down at about step 7. So the 1,3-bisphosphoglycerate down there. And so see where it gets turned into the 3-phosphoglycerate. So it produces a molecule of ATP there with that enzyme. But with the Rappaport-Lubring shunt, instead of going there, it goes down to a different thing. So instead of going down the phosphoglycerate kinase pathway and generating an ATP, it goes down this shunt or side path. Now, can you tell me, yes, from that, what it produces as part of that? And does that look familiar to you? It has a few different names, which makes it confusing. That's the problem with a lot of these metabolic kind of groups. They have multiple different names, but.
So the one in the middle, the 2,3,5-phosphoglycerate is also what we know as 2,3-DPG.
Yes, that's right. So sometimes they'll swap a bis for a di, kind of means the same thing. Probably one's Latin, one's Greek. So yeah, 2,3-DPG. And so this is how it gets produced. And so, of course, yeah, as anaesthetists, we're interested in its effect on the hemoglobin dissociation curve. And so this pathway is found predominantly in erythrocytes. And the interesting thing about it is that it's what's called a futile cycle. Like it actually, in order to do that, it sacrifices the production of an ATP molecule. So it's kind of not without cost to produce that. And so, yes, that's just a little side note that probably worth knowing, that that's how you get the 2,3-DPG that we're interested in for other reasons. Now, how are we going? So it's nearly 5 o'clock. So yes, this is just looking at other potential sources for the acetyl-CoA. So again, coming back to that central molecule of acetyl-CoA in the middle, that then goes through the citric acid cycle and produces all that NADH that gets used in oxidative phosphorylation. So this is where all the amino acids kind of come in, and they talk about so there's ones that are kind of glucogenic, so the alanine, cysteine at the top, they kind of been turned into things that are part of glycolysis or can go into glycolysis. So in theory, they're the amino acids that can be utilized in gluconeogenesis. You can start breaking down proteins during starvation or fasting. You can break down proteins to turn them into some form of glucose that you can use. And they're the ones, the glycolytic kind of amino acids, whereas the others are called, they call them ketogenic, because they feed into acetyl-CoA that can then form ketone bodies in starvation. So they're the other ones, and some of them can do both. So again, yeah, this is just, again, highlighting the central importance of acetyl-CoA and how the amino acids can end up being incorporated into that, but also gluconeogenesis. This one's, so this is beta oxidation. So this is how fatty acids and lipids can get converted. And again, you'll see up on the top left there, kind of acetyl-CoA kind of being produced. So this is kind of, you've got a long chain fatty acid, and through each cycle, two carbon molecules are kind of broken off that long fatty acid. It goes through this beta oxidation cycle, and in the process producing acetyl-CoA. So that's kind of fats and lipids, and another source of acetyl-CoA. But again, once you've got the acetyl-CoA, you're stuck with it. You can't kind of shoot that back up and produce carbon dioxide, glucose again. So there's plenty of options for us producing acetyl-CoA, which can then go into the Krebs cycle and then into oxidative phosphorylation. But it's, yes, none of that is any good to us when we haven't got enough glucose. And none of it is of any good to us when we're in anaerobic conditions. And this is, yes, probably the final slide. So this is, yes, my hand-drawn summary of all that kind of metabolic stuff, which again, kind of cover in a bit more detail in a future tutorial. But I keep meaning to make it into a friendlier infographic. But this just, yes, highlights that central role of acetyl-CoA in the middle there, and then where all that fits in, because, yes, aside from the things that we've discussed of going into the tricyclic acid cycle, and then through that into oxidative phosphorylation, it's involved in so many other pathways as well. So production of cholesterol, ketone bodies during starvation, even acetylcholine is created from acetyl-CoA. It's produced from amino acid metabolism as well as free fatty acids and so on. And yes, it's a bit hard to see, but that the straight line running from the left of screen then up and across, that's kind of representing the mitochondria. So all the acetyl-CoA tricyclic Krebs cycle, all that is happening inside the mitochondria, whereas the glycolysis, the glucose metabolism, is happening outside in the cytosol. So that's probably it for me. I'm happy to go back through some of the previous slides or take any questions now. Oh, I might be able to. I still don't know how to. I might, if I stop that, I might be able to get my. Oh, there we go. I think I'll be able to. Go into. yeah, so if anyone's got any questions, can shoot them to me or...
I just had one small thing.
Yep.
So in anaerobic metabolism, because of the lack of oxygen, the only way to generate energy or ATP is through glycolysis and the Krebs cycle, and then
not,
using any other substrates to feed into those.
oh, sorry, go on.
No, that's all. That's all.
Oh, sorry. I thought someone else was jumping in there. So yeah, your question was during anaerobic conditions, you've only got glycolysis to produce ATP. That's correct. But yes, not the Krebs cycle. So so if we go back, so yes, the Krebs cycle is based on acetyl-CoA. It started by acetyl-CoA, and it does produce some ATP. But the problem is that because you've run out of oxygen, effectively, this isn't working. So the whole electron transport chain backs up because you haven't got the oxygen to offload the electrons at the end of it. And so you're stuck with NADH, but you don't have any NAD. And in the Krebs cycle, the citric acid cycle, you need that NAD plus as well. So that shortage of NAD plus brings the citric acid cycle to a halt as well. So you're not even getting the GTP/ATP from that as well. So you're solely dependent on glycolysis in those anaerobic conditions. And this is happening in the cytosol rather than the mitochondria. And so you need that NAD plus, and the only way you can get that back is through converting the pyruvate that's at the end of glycolysis, converting that into lactate that kind of regenerates the NAD plus, and the lactate then goes on to either build up and cause problems or gets metabolized by the liver or the heart, or turn back into glucose depending on what the conditions are. Does that kind of answer your question, or have I misunderstood it?
No, that answers the question. So then, following on from that, if the lactate if some of it does get reconverted back to glucose, then that whole Cori cycle sort of thing with pyruvate just keeps going around to make the limited ATP that you can.
Well, yeah. So I think, and this is why anaerobic metabolism is such a nasty thing, and like we can maintain it for short bursts, like during intense physical exercise and those sorts of things, but it's you're not going to last long doing that because all the kind of systems like the oxygen debt and the lactic acidosis kind of builds up, and in the pathological sickness state, it's kind of really bad because yes, while in theory, like I think if it's in kind of I think that I can't remember exactly what the pathways are for gluconeogenesis, but I think that conversion of pyruvate to lactic acid as a means to continue glycolysis is just a mechanism to buy you time. Like, I don't think that the Cori cycle kind of happens on an adequate time frame to kind of maintain anaerobic metabolism for long. And I think that you would need, you'd probably run into the same issues with running out of NAD plus for that kind of, well, I think with it gets a bit more complicated with gluconeogenesis, because then I think it's NADPH, which is a slightly different molecule. But yes, I don't think the Cori cycle is a mechanism of maintaining anaerobic metabolism. It's more of a mechanism of generating glucose during times where during times of starvation or fasting where you're not getting adequate dietary glucose. And yeah, I can't remember off the top of my head the specifics of the Cori cycle, but yeah, probably cover that a bit more in the metabolism shoot later. So yes, I think that the pyruvate to lactate is just a very short-term mechanism within a particular cell to keep the local concentration of NAD plus adequate to keep glycolysis going. And yes, as we know, if it goes on too long, then it starts causing problems. Is that helpful or further clarification?
Yeah, that's fine. Thank you.
Anything else? As I said, yeah, so this has been recorded, is that right?
Yes.
Yeah, this is being recorded if that's okay with you.
Yeah. No, absolutely. Yeah, I just wanted to ask. So, yeah, I guess if you guys are happy for me to add it to the blog site for future reference, is that?
Oh yeah, sure thing. I can CC you in an email I sent to Bianca, and she can send it around.
Yeah, that'd be great. And yes, and yeah, if you have any questions afterwards, or and yeah, so links to some of the articles that I've referenced, or will be on the blog site as well. So yes, if you go there or have any other questions, yes, shoot me an email and I'll see if I can help you.
Hi, just from Tamworth, how do we come to these blog sites?
Yeah, so if you go into we'll go back to the, so I think, well, on the anesthesia training website, do you have the details for that Sasha kind of runs? So there's a link to it there.
There you go. I think Paul's put something up.
Yeah, and I've put it in the chat for this Zoom meeting as well. So you should be able to click on it, and that's that's it there. And that's got just various references and resources that I used in my primary exam many years ago that may or may not be helpful to you, but it includes the slides and audio from the tutes that I've done in previous years. Of course, yes, I keep thinking that I'll just repeat it each year, but inevitably I end up changing it a bit. And also different years, different topics will be of interest, or different questions will be asked. So it does vary a bit each year. And of course, this year is very different to previous years as well, being Zoom. Normally I try to make them a bit more interactive and stuff like that. So yes, you're welcome to go and take a look at that. And
thank you.
Yes.
And I'm so sorry. This is a very might be a very stupid question, and it's not a Tamworth question. It's just a me personal question. So it doesn't reflect the high IQ of Tamworth. But in terms of oxygen, apart from being involved in the oxidative phosphorylation in the electron transport chain, where is it used?
My understanding
are there any enzymes or like other things that that uses oxygen? Because I often see like water being a part of like a reduction, like, you know, as a factor for like as a reducing agent. But is oxygen used anywhere else? I'm so sorry.
Yeah, no, that's a very good question, and one that I've not really thought about too much. But my understanding is that probably no. Like I think that's would certainly be, oxidative phosphorylation would be where the vast majority of the oxygen that we breathe in and gets used is used, because the pretty much wherever there's the only cells that don't have mitochondria are red blood cells, and so they're completely dependent on glycolysis for their ATP, but then they're not that metabolically active. So I may be wrong, and there may be some small, kind of niche enzymatic reactions, or there might be oxygen that's incorporated into some other structures, but No, I think, yeah, most of the other oxygen that gets incorporated through gluconeogenesis or other kind of catabolic, anabolic processes probably come from water. Like I think, yeah, water and the OH molecules are probably a much more easy to use source of oxygen for producing glucose or fats or proteins than getting it from oxygen, but I could be wrong. So yeah, no, that's an interesting question, not a stupid question at all. Does anyone else can think of any other metabolic reactions that directly involve molecular oxygen as opposed to like water or CO2?
The only other thing I can think of that's not necessarily good is just oxygen-free radicals, but that wouldn't be molecular oxygen.
Yes, no, that's interesting. Yes, that's a good one. Yeah, and I'm not even sure, yes, how that comes about being generated now. Like, yeah, whether that's from a splitting of an oxygen molecule or from dissociation from some other oxygen-containing molecule that was generated by other means, I'm not sure. Yeah, I might have to look that up for next year. Anything else? No, maybe wrap it up there. Again, I've lost my mouse pointer, so I can't. Maybe I'll get rid of that. I think that will do it.
Thank you very much for the tute today.
Oh, not at all. My pleasure. And I think there'll be a few more coming in the next couple of months as well. So when's the next, yeah, what's happening with primary exam sittings and everything?
Still going ahead on the 18th of August.
August, right. So that's...
written, that is. And Dan's got his Fiver coming up in October.
Oh, right. So did they get postponed? Was that the Vivers?
Yeah, we're sitting the Vivers at the same time in October.
Yeah, right. Okay. Well, yes, interesting times. Well, good luck with your study. And yep, as I said, drop me an email or something if you have any questions or anything like that. And catch you later.
Thanks very much, Reese.
See you later.
Thanks.
Thanks, Reese. Bye.
Thanks, Reese.
Thank you.
Thank you.
Thank you.